before we start with this PBO, placebo beyond opinions, well, it is our center, School of Nursing, the University of Maryland. We are committed to moving beyond more opinions or assumptions related to placebo and placebo research. Our center focuses on advancing beliefs, claims about placebo with obsolete and unbiased lens. The goal is to understand evidence-based inquiry research mechanism and translational research related to placebo. To this goal, we invite this year 3 speakers. So one is here tonight with us, Dr. Massieh Moayedi from the University of Toronto. We will have another speaker in a few weeks, Stephen Cohen from Chicago, and someone from NIH, Massieh Moayedi. So we welcome all of you to this series this year, and we're very excited to start with this. Thank you very much for being with us. Our co-director of the Placebo Beyond Opinion Center will introduce our speaker.
Thank you, Luana. And okay, so it's my pleasure to introduce Dr. Moayedi, who's here today from the University of Toronto. He serves as associate professor and co-director of the Center for Multimodal Sensory-Motor Pain and Pain Research. His expertise is in pain neuroimaging, including structural and functional MRI, including working with vulnerable populations. His approach to research has been to study basic scientific questions in both healthy participants and in clinical populations, allowing him to really have the patient's experience shape the focus of the research. He holds grants from CIHR and CERC, CFI, and NIH. He's on the editorial boards of the pain and pain reports journals, and an associate editor at Frontier of Pain Research. He founded the International Human Pain Seminar Series and serves as founding chair of IASP Neuroimaging of Pain, SIG. The presentation today promises to be a very exciting one. It's titled Contribution of Endocannabinoids to Pain Sensitivity and Modulation. Thank you very much.
Hi, everyone. It's an honor to be here today. I spent a lot of time in Maryland during my postdoc, and I worked really closely with Dave Sminowitz. So coming back here and having the opportunity to share some of my work is really great. So I'm going to talk to you about a few projects we're working on in the lab. Most of the data I'm going to show you is actually unpublished and is brand new. Some of it is literally off the server from last night, but it's exciting and I thought I'd share it. So let's dive in. Just disclosures, because I think it's important. I basically have no financial conflicts. I have funding from various federal funding agencies in Canada and NIH, and I have various roles in various societies related to pain. So what is pain? Most of us know this, but I think it's good that we're all on the same page to begin. So pain is by definition an unpleasant sensory or emotional experience associated with actual or potential tissue damage. There's the older definition versus newer ones that are slightly different. But ultimately, I think it's important to understand that pain is in the brain, right? So the nociceptive system carries pain to the brain. Oh, oh no, that did not work. Okay, maybe just do that. Okay. So ultimately, pain is in the brain, meaning that although we have nociceptive signals coming in and we have modulatory systems modulating those nociceptive signals, the experience of pain is bound in the brain. It has to come together to create a more complex experience. And it is shaped by our lives, by our genetics, and our social circumstances. So we know that there are individual differences in pain, right? So what might feel like a light touch to someone might be excruciating to someone else. We often struggle to understand that, right? We try to center the way we interact in the world and the way we understand other people's experiences through our own experiences. So it makes it really difficult to assess other people's pain. But ultimately, there was a study as early as 1944 that showed the number of subjects who would report pain at a 5 out of 10 across these various temperatures. So what this is at the bottom is calories of heat per centimeter of skin, which is a very outdated way of giving heat to the skin and measuring it. But effectively, what you see is that there's a huge spread of the ratings. More interestingly, there's this paper that Roger Billingen pulled out and highlighted in his review, where 321 people were given a 48-degree stimulus, heat stimulus, and the ratings vary from 0 to 1. Right? So it really shows you the breadth and spread of pain that people experience. And that can be measured through quantitative sensory testing, which are techniques that quantify the pain experience in individuals. But we can also capture that individual difference using brain imaging, right? So we know that there's evidence that individual differences in pain can be captured through univariate and multivariate patterns, and that the brain ultimately does encode individual differences in brain and pain across participants. So we wanted to look at whether we could understand what systems contribute to that individual differences. So we used a novel ecologically valid model of orofacial pain. And the way we did this is participants would come in for a baseline resting state scan. There were 26 individuals. And then we did what I think is a terrible pain model in that it actually really hurt and sucks. But basically, it's a standard orthodontic process where you put an elastomeric separator between the teeth. And basically what this does is it pushes apart your teeth and causes inflammation in your jaw. And what's nice is that it peaks at 24 hours after insertion. So we put it in and we send them. And what that allows the participant to do is live their lives, right? And we get pain measurements through text messages 3 times a day. And ultimately, it is a pain model where it's not in the lab, it's not artificial, it's really annoying. And you can calibrate the amount of pain you get by changing the thickness of the separator you put, where someone's putting in 2 separators. And then we try to find the brain regions associated with this. So as you can see, we had a spread in the pain ratings across individuals. On average, the pain is low. That's because we didn't want people to pull out the separator overnight if they got too bothersome. But you do get a nice spread of pain over 5 days. But again, in most people, it does peak at around day 1 or 2. When we looked at the brain, what we found is that the laminocortical networks from the thalamus to the insula bilaterally predicted how much pain someone would have, quite robustly actually, 62, R squared of 62 or 62% of variance. And then on the other hand, the top-down processing was associated with the connectivity of the default mode network with the periaqueductal ray. So a region known to be involved in the semi-maculatory system. And what's interesting is that when we did a model to see whether these captured unique variants, they did. So additively, they actually captured much more variance in the pain experience than not. So this brings us together to bring these 2 concepts together that, bottom-up processing and top-down processing do shape the pain experience. And that's really what Irene Tracy and Ulrike Winkel were highlighting in this paper. And that we've known for quite a long time that these 2 systems do work together. But how are these signals shaped by neuromodulators? And what I'm going to do now is talk to you a little bit about endocannabinoids. So the endocannabinoid system, I don't know if you can see my cursor, but I will pick one if I can. Yeah. So the endocannabinoid system is an endogenous system that comprises of the CB1 and CB2 receptors. Here you're only seeing CB1. The CB1 receptor is distributed in most tissues in the brain, is one of the most common receptors in the human brain. And what's interesting about it is that we have ligands that attach to them, and those ligands include the 2-AG, which is broken down by DAGL, or formed by DAGL and broken down by MAGL. But then there's other ones, anandamide or AEA, that is broken down by FA. And we have two other molecules that aren't strictly endocannabinoids, but they also are broken down by FA. And so there's a substrate competition for the enzyme, and that's OEA and PBO. I'm not going to try to say the names because I am not a pharmacologist and I can't. But ultimately, what we do find is that these four molecules are suspected to be involved in pain. One of the reasons we suspect that is because, surprisingly, AEA binds not only to the CB1 and CB2 receptors, but also to TRPV1 receptors. And TRPV1 receptors are heat pain receptors or noxious heat receptors that we think encode noxious heat, and well, we know encode noxious heat and can lead to heat pain. So, so that gets quite interesting, and come together, it gives us a system to look at whether or not they modulate pain. And that's kind of fun because one, there's been this large focus on opiate receptors and opiates in the pain system. And we know the detrimental cause of that over time. But one of the, as cannabis gets more and more legalized, we still don't know the effect of cannabis on pain. And ISP released a series of task force papers that really did show that there just isn't enough evidence to understand the relationship of cannabis and cannabinoids in pain, which is really what led us to do this. So the relationship between pain and the cannabinoid system comes from this, well, this is one of the pieces of evidence, and I'm sure you all saw this story, and the title of this paper really doesn't give it away, but ultimately there was a woman who felt no pain when she gave birth, and she really didn't understand the cause. And that's because the endocannabinoid system is involved in anxiety regulation and stress regulation, as well as we think pain. So this is, a really nice, easy way to understand that this system is involved in pain. Similarly, we know from rodent models that that this same deletion in rodents can lead to differences in the concentration of anandamide in the brain and in the blood, and that affects the way that they respond to heat pain. So here, the rodents are left on a hot plate, and it's how fast they respond. And you notice the latency is longer in those who have basically single nucleotide polymorphism. This is also true for tail immersion, the hot plate, and the latency for licking for the formalin test. So ultimately, what you do see is that there's reduced sensitivity to pain in rodents who have a FA polymorphism or FA gene polymorphism. So we also know that the CB1, CB2 receptors are distributed across the cortex. There seems to be a higher affinity for AEA and CB1 than CB2. AEA also does bind to TRPV1, as I said, and 2-AG binds to CB1 and CB2. And so we see them in the periphery at the primary afferent level. We also see them in the dorsal horn. They play a key role there. They're quite well distributed. And interestingly, we also see them very distributed in the brain, particularly in the thalamus and in the periaqueductal gland, two regions that we know are quite important for nociception and pain modulation. And they show the microslide. And then they're also quite distributed in cortical regions. And so the question is, how can we leverage this to start managing pain, and how does that work? And the issue with the endocannabinoid ligands is that they're very short-lived. They're really created on demand, and then they're broken down soon after. So they really do exist very briefly in the nervous system. So there's been this focus on enzymes involved in breaking down anandamide. Not so much the one for 2-AG, which is vagal, but thought has been really a loci of focus. And I think it's really related to those studies that we saw about congenital analgesia and without many consequences, right? She lived a normal life, she had kids, she aged normally. So it's very different in the congenital analgesia you see in individuals who have like a sodium channel receptor polymorphism. It really is a different type of congenital analgesia. Nonetheless, so this gene got really popular. It was studied. For placebo by the Zubieta group, and ultimately what they found is that people who have the gene polymorphism or homozygous for the gene polymorphism reported higher placebo analgesia and more positive affective states 24 hours after placebo administrations. But there was no effect on pain in the absence. So it's really interesting because they seem to modulate pain in the presence of a modulatory profile, but not in the absence of it, which is not quite clear to me why, but I think it's an interesting fact. And you can see here that relationship. So ultimately, this is change in pain using the pain questionnaire versus the amount of opioid in the thalamus after placebo administration. And you can see that that relationship is quite tight, but it's probably different for the two groups. Right. So we also, there's a study by Benedetti that also showed that placebo analgesia is mediated by opioid and non-opioid receptors, and specifically he's talking about the CB1 receptor here. So, so Benedetti used rimonabant to understand whether someone primed with opioid analgesia and then placebo, for someone who had primed with ketorolac and non-opioid analgesia, you see different outcomes. And ultimately what you see is that in the presence of rimonabant, the placebo response is significantly blunted after ketorolac, but is really not changed after morphine. Although this study does lots of statistics, so it's hard to really interpret the data. So all this led us to designing a study. This study was led by my student, Rossi, who you can see here. And we collaborated with Lauren Atlas and Dave Finn from the National University of Ireland, or University of Galway in Ireland, and Lauren Atlas from NIH here. And this was quite a complex study because it was really long, as you can see. So participants would come in and they were fasted. And then we would give them, we do a screening for BDI, no depression, pain responses, and potentially endocannabinoids. So we screened against clinical levels of BDI and suicidality for safety. And then we took a saliva and blood draw. And then we did a calibration. And then we did a placebo manipulation. Following that, we also did an offset analgesia and a conditioned pain modulation paradigm. One of the things that we did in this study that many other studies haven't done yet is we collected blood before and after each procedure. So as endocannabinoids are fabricated or produced on demand and broken down quickly, it's important that you can get those dynamic measures rather than static measures. So we think that endocannabinoids are not trait-like, but are more state-like, whereas the enzyme, the ligands, whereas the enzymes are more trait-like, right? So there's a genetic difference between the PBO gene, and that can moderate various pain responses. But the ligands themselves are different. So this is what the blood draws look like for the 4 molecules of interest, the 4 ligands, AAA being anandamide, 2-AG. So these are the true endocannabinoid ligands. And then these are the NAs, PA and OA. So not strictly endocannabinoids, but they do compete for the PBO gene, for the PBO enzyme. So what you see is that there's a clear pattern of change that occurs here. And that is mimicked here in AEA. And so what that tells you is that AEA, and we know this, that AEA, PEA, and OEA are highly correlated in concentration, whereas 2-AG is its own thing, and it's very different. And so what we did first is we used Lauren Atlas's approach to find regions on the forearm where you can get a stable rate. So it's really important that when you do a placebo paradigm, that you're comparing like to like. And so Lauren's developed this very complex and rigorous approach to give multiple stimuli over and over again across 8 sites in the forearm to try to identify regions that will produce a stable response with multiple stimulations and that are comparable between sites. And so we did this, and we used this pain scale from no sensation to the most pain imaginable with a few anchors that are different than standard VAS, but I think is more intuitive for participants to use. Once we had the regions where we knew we could test, we went ahead and did a standard placebo conditioning effect. So this is a standard one where you tell the participant that they're getting a placebo cream on one patch of the arm versus a Vaseline cream or control cream on the other. And then you condition them by giving them different temperatures on the site, right? So you give them a 4 out of 10 pain here and an 8 out of 10 here. And you tell them that the cream did the same and that the placebo cream is very powerful, and then you give you rating. And then when you do the test, what you do is you either test the placebo cream or the Vaseline cream, and you counterbalance the order in which you did this, but we did both. And effectively what you get, but you give them the same temperature. And what's interesting is that you get vastly different ratings on those regions. So what you've done is built an expectation that the blue squares here, or this placebo cream, is really anti-nociceptive and will hurt less, whereas this Vaseline control cream doesn't protect against it and will hurt more. But when you give a pain stimulus that's between those two, you tend to get very different ratings. And that's exactly what we saw. We saw that the placebo site, people rated pain in the placebo site significantly less than they did in the control site. And this was true for most participants. Not everyone is a placebo responder. We do have some nocebo responders, so in the placebo site, we actually gave a higher pain examples. So the placebo in our case that I'm going to show you from now on is operationalized as control minus placebo. It's a little bit counterintuitive. We just did it this way for fun, mix things up, I guess. But ultimately, what we do is we did control minus placebo. So keep in mind that the analgesia metric we have for placebo analgesia is really the other way around. So the first question we wanted to ask is, do baseline endocannabinoids and NAEs predict placebo analgesia? And we went in with a negative expectation of this, or we just didn't expect it to work out. And the reason that that's the case is because, again, endocannabinoids are created or fabricated or produced on demand. So it would be odd for baseline levels of endocannabinoids and NAEs to predict a pain response. Now, the exception to the rule to that would be if you're looking at different genotypes for the progeny, right? So those who have the polymorphism for the progeny that allows ADA to stay in the blood longer in PA and OAA as well. We didn't see that relationship here. These are some of the most zero-order correlations I've seen, but they're there, and they're quite robustly flung. But are changes in ECBs and NAEs associated with placebo analgesia? So in this case, we know that we have 4 ligands, 3 of which are highly correlated to each other, and you can see that here. ADA, PA, and OEA differences pre and post placebo are very highly correlated. So what we did is we basically ran a principal components analysis, and as we expected, we got 2 principal components, the loadings of which are quite obviously robust. And then, so these are the 2. This one is entirely 2AG with some contribution from ADA and OEA, and this component is entirely 3 with no contribution. And so, did it predict? The AEA-OEA the AEA-OEA component was correlated with the placebo analgesia. And again, remember that this is the other way around. Ultimately, the more endocannabinoids you have, the more analgesia you have. So that suggests that endocannabinoids and NAEs certainly play a role in placebo analgesia. 2-AG, however, again, is zero order correlation to as we would expect. What's interesting about this ultimately is that this doesn't tell us yet whether the progeny is involved or not, but we would expect it to be involved. So the next step of this is for us to genotype these individuals and see whether or not this relationship is moderated by carriers of the polymorphisms, and whether that's tiered in the way that we expect. So stay tuned. The next test that we did was offset analgesia. So offset analgesia, for those who don't know, is a really interesting phenomenon that I don't think we fully understand. There are some theories out there, but I'm not convinced by them. And ultimately, what you do is you give a noxious heat stimulus that's moderately painful. In this case, this participant rated it a 7.5. And then you raise it by 1 degree for a brief window of time, about 10 seconds. In this case, we did, I think, 5 seconds. And then you go back down to that 46. And what you see is that as you increase that temperature, the pain goes up, the pain ratings go up. So in black here, you see the individual's pain ratings, and in green, you see the temperature of the thermal. And the pain goes up, as you would expect, as you raise the heat. But when you drop it, you see this profound analgesic effect that lasts for 30, 40 seconds. That profound analgesia is, in my opinion, quite remarkable. If you've ever done this paradigm, no idea if you haven't, you should try it. But ultimately, it's one of those things that we don't fully understand, but is quite cool, frankly. And I'm really interested in it, and I'd like to unpack it some more. But the question being, is it related to OEA, or endocannabinoids and NAEs? So just so that you can see first, we had a robust offset analgesia effect in those individuals. We had 2 non-responders. And when we compared, what am I trying to say? When we tried to see whether or not they correlate with baseline, we were surprised to find that AEA is actually a robust predictor of offset analgesia. So in this case, we have a trait-like effect, right? This is when they've come in, first blood draw they've done, they haven't undergone any manipulation, they fasted overnight, and we draw blood, and it's a nice correlation that predicts whether or not offset analgesia occurs. Now, I was really skeptical about this finding, to be completely frank. So I ran a Bayesian-Pearson correlation as well to get a Bayes factor to tell us how much evidence there is in the data to support this finding. And again, much to my surprise, the Bayes factor showed strong evidence in support of the correlation. So we now know, whereas for for OEA, and this should say OEA, for OEA, where there was a modest correlation, there actually wasn't a BF10. I showed the wrong statistics, but I guarantee it was not supported by the Bayes factors. And here you can see that correlation at baseline. So this raises a lot of questions. One, why is offset analgesia predicted? Why was at baseline, whereas placebo wasn't? And I just don't have the answer for that. And that I think warrants quite a bit more research. But we then wanted to see whether dynamic changes in endocannabinoids and NAE were associated with offset analgesia. And again, we did our principal components analysis to reduce the number of multiple comparisons and to reduce the linearity that we have in our regression. And ultimately, we have one component for the three endocannabinoids that change before and after offset analgesia, and we just use 2-AG on its own, because we really wasn't. So what we found is no correlation. So in 50 individuals, 49 individuals, 24 females, 25 males, you do not see an effect of endocannabinoids predicting offset analgesia. The one thing that I haven't done here and that I think warrants maybe further analysis is to see if ADA alone is predictive. Although the loadings on the components here are quite high, but a little lower for ADA. So it's possible that this unique variance here in this component might actually be what's driving that relationship, and that warrants further analysis, in my opinion. And then this is basically what we did find in our regression model. It was a clear sex difference. So there was a significant sex difference in our model between males and females for offset analgesia, but this was unrelated to females. So again, stay tuned for future analysis. The last paradigm that we ran was condition pain modulation paradigm. And for those who aren't familiar, this is also known as counter-medication or various other titles. It's thought maybe to be the human equivalent of DNIC or diffuse noxious inhibitory controls, but there is some debate about that. We do a slightly different condition pain modulation, which is, I think, more tolerable. So instead of doing a conditioning stimulus, what we do in conditioning stimulus is just cold immersion. So you stick your foot in an ice water bath. And then what we ask you, and then what we do is we do a pressure pain threshold before, during, and after. And so instead of doing a super threshold noxious stimulus, what we do is we try to see whether the thresholds change. We do this for two reasons. One, I tend to think that deep pain is more ecologically valid in general and is more realistic, as opposed to cutaneous heat pain in a laboratory setting with a safety standard. We're not going to do it. In this case, muscle pain is muscle pain. You press on it, it hurts, and it's easier for people to also report it hurts. Nonetheless, we ran this model. Interestingly, and this is a total sidebar, but I find it really cool. If you go back to Hippocrates, he was the first to actually write down that DNIC existed. We talk about counter-irritation in that person. So it is something that we've known about for a long time, but we've only started talking about that. Sidebar, but I think it's good. Nonetheless, CPM was effective. So you see an increase. You see the pressure pain thresholds increase during the immersion, suggesting that there is a analgesic effect. So their pressure pain thresholds are higher, they're less sensitive, and that it goes back to baseline or no different than baseline after immersion. So the difference with this model, of course, is that it's not as sensitive to post-immersion CPM effects. And but I think it's one, better tolerated by participants, and two, more ecologically valid. But what we found is that baseline NA did not predict CPM whatsoever. You can see that here in the article. So again, a bunch of zero-order correlations, very close to the correlations. Again, we don't have the FA gene type just yet to see if whether that somehow moderates this relationship or shifts this relationship if we control for it. But to be frank, I don't expect to see that. So again, we have to break down our variables. And so we have two components again, they're very nicely loaded separately. And when we look at whether or not ECVs and NA are associated with CPM, the answer is no. In fact, the 2AG is not at all associated. There's a very slight relationship, but it's not really significant in the AEA, PEA, or relationship. And sex didn't matter as well, either, which is a bit different than what we know in the literature. CPM is thought to have sexually dimorphic effects, but it didn't come out in our sample. And it could again be that we use pressure pain threshold instead of super threshold noxious. So I think what I've shown you so far is that baseline endocannabinoids and NAs do not predict placebo analgesia or condition pain modulation, but baseline AE did surprisingly predict offset analgesia. Placebo analgesia is associated with changes in AEA, PEA, captured by a single principal component. And that's not surprising that three of them would be related, given that they compete for the they have substrate competition for the PA enzyme. Oxide analgesia was not associated with changes in endocannabinoids, and conditioned pain modulation was not associated with endocannabinoids or NAs. So it's an interesting story that I think makes us think a little bit about what's going on. I think what we can take away from this is that placebo analgesia clearly has a endocannabinergic component that is dynamic. Conditioned pain modulation does not seem to have any endocannabinoid, endocannabinergic contributions. And oxide analgesia, I just couldn't tell. I don't understand it. So again, stay tuned. I'm going to talk about one last line of research that we're doing in the lab in collaboration with Isabelle Boileau. And I think this is a really exciting line of research that I got to do. But let's get into it. So this is a study in the early days of discovering the PA gene has polymorphisms. This was like the age of Elon, in 2002, genetics was all the rage. And basically what you find is that people who have the SNP, so the C385A polymorphism in the PA gene, and are homozygous for that SNP, basically have higher percentages of substance use disorders, whether it's drugs and alcohol, or street drug user and drug alcohol. And so that's quite interesting, right? Because these are individuals who have, based on what we know about the PA gene, greater amounts of endocannabinoids circulating in the blood or in anti-PA motivating. And in that one woman from Scotland, she had less anxiety and less pain. But they seem to also be coinciding with greater substance use disorders. I think that's not as intuitive as one would think, but I think it's an interesting finding. Isabelle works on PET, or positron emission tomography, and this is, for those who aren't familiar, it's a brain imaging technique that allows us to put radioligands in the blood that then bind with specific receptors or ligands in the brain. And in this case, it binds with the FOB enzyme. So the curve is a radioligand that is designed to bind with FOB. And what you see here is basically that binding affinity throughout the brain. And like I said, endocannabinoids are very present in the brain. They're very well distributed, and ultimately that's what you see here. So Isabelle did this study, is doing this study, and I thought I'd show you some preliminary data that's fun, where she's comparing people with opioid use disorder and chronic pain, people with chronic pain without opioid use disorder, and people who are health control. So OUD plus is chronic pain plus opioid use disorder. OUD minus is just chronic pain. Wait, no, that was backwards. Sorry, this is opioid use disorder with chronic pain. This is opioid use disorder without chronic pain. These are healthy controls. We don't yet have, in this data set, the chronic pain only cohort. But ultimately, it's a small sample for the two patient groups. These are not easy patients to find. And when you do find them, many of them are not interested in undergoing a radioactive brain imaging scan. Nonetheless, I went up to Isabelle and I told her about what we're doing in our lab, looking at endocannabinoids, pain sensitivity, and population sensory testing and imaging, and she got quite excited about that. And we decided that maybe we should try doing UofT in these patients. And so first I asked her to send me the distribution of FOB in the brain. And what you find is that that distribution varies a lot based on the genotype of the FOB. And that's surprising. So those who are heterozygous or tend to have more distribution of the PA gene in the brain. Don't know what that. Whereas those who have homozygous for the polymorphism tend to have less distribution of the PA gene. Why that is, I'm not sure. Why you would have fewer binding affinity of the enzyme, I'm not sure. I suspect it's because CURB binds to the PA enzyme, and that when you have the polymorphism, it doesn't bind as well, but I'm not sure. and that's a question you can ask you. Nonetheless, we took the expression of FA for each individual in each brain region, and we correlated it with our battery of pain sensitivity testing. What you find is that in general, innocuous thermal and mechanical stimuli are associated with the distribution of FA and quite robustly. Again, very small sample size, right? So let's not overinterpret this. But there seems to be a relationship there. In thresholds, that doesn't come out for cold pain, but heat pain and mechanical pain, those relationships will be there. Similarly for pressure pain, and then the cold water pain threshold. So that's when you stick your hand in an ice bath and you see how long you can keep it in before you pull your hand out because it hurts. This is the threshold, is when you first say it hurts. So interestingly, the thermode-based cold pain the threshold is not related whatsoever, whereas a cold water pain threshold is. And if you've experienced these two different tests, they're very different. One is really overwhelming when it gets cold, and the other one is fine. Some people can tolerate it down to the lowest temperature and they're fine. But if you stick your hand in an ice bucket for 30 seconds, you're going to want to cool down very quickly. Similar to what we saw, conditioned pain modulation doesn't really show much of a relationship. Temporal summation of the test we did here, which measures the ability of the spinal cord to sensitize, also not related, which was surprising because these are where we think the opioids would have, not the endocannabinoids would have the most effect. So that is something that we can't really square with our understanding, but again, we could be wrong. And then here the cold presser task is where they stuck their hand in the ice bucket for as long as they could. And that again was very highly important. So I think what we take away from this is that, and various other studies that I haven't shown today, is that endocannabinoids are recruited in as needed. But I think what we've learned is that you actually need to have quite a stressful, noxious event. Threshold events or light pain events. And even some laboratory pains where there isn't that much stress involved, there isn't that much ecological validity to it, aren't related with endocannabinoids. So I think that these are early days in this field of research, and there's a lot left to do, and we're really enjoying this. So just to sum up my talk, pain is a dynamic process that's shaped by bottom-up, ceiling, top-down modulation. Pain modulatory paradigms differentially engage the endocannabinoid NAE systems. We see that placebo analgesia seems to be related to the FA gene and its polymorphisms. And offset analgesia is only related with baseline AEA. And I wonder in that case, is it a FA-related trait effect? In that case, the FA gene is what's driving the differences in offset analgesia, and that's why the dynamic effect doesn't matter. But again, early days, we don't know. And my last sentence is not complete, but we think that FA is associated with measures of pain sensitivity and brain regions that encode those processes. I'd like to thank my lab and my collaborators. This is my wonderful lab, who are incredibly hardworking and lovely people. This work was led by Rossi and supported by Lauren and Dave Finn. And Kevin, I want to say a huge shout out to because he has been spending the last 5 days quantifying our endocannabinoids. So when I say this data is fresh, literally I got it yesterday. But I think they're quite exciting. So when I say it's early days, I mean it's early days. There's a lot to unpack here. But I think when I saw the data and I saw what's coming out of it, I got very excited and I thought I would share that with you guys today. This work was entirely funded by the Canadian Institute of Health Research, so I have to give them a shout out. And thank you.